things I kind of brought up last time is the reason we sort of care about affinity and specificity as why do we care about the thermodynamics of these binding reactions is essentially to inform drug design. Okay, so we can think about how temperature would affect these things. We can think about how different concentrations would affect the binding and sort of come up with a system that would work when we want to do rational drug design. Okay, I decided to keep a handful of the slides from the back half of chapter 12 in, but a lot of them have been removed. I put them up last Wednesday, so if you look at last Wednesday's and then you look at today's set, you see the ones that are now missing. Those are the ones I won't ask you any questions about. So the drug development process, I did go through this really quickly last time. So it starts with sort of basic research or just trying to identify what proteins and pathways are even involved with the disease to begin with, which this alone can take years and years of research, okay? So assuming though that you identify some protein or enzyme that's involved in the progression of a disease, some enzyme that's going off the rails and doing something you don't want it to, then you want to inhibit its function. Okay, this is sort of a typical setup. You have an enzyme that's performing out of control, it's activated when it shouldn't be, nothing's bound to it, and it's still initiating its cascade of events. So you want to inhibit that protein, and generally there are two sites that you might target. The active site, so this is the site where that enzyme or that protein's normal ligand binds, okay? So if you target the active site, you put your drug there such that the actual ligand can't bind anymore, okay? Alternatively, you could intervene at an allosteric site. So this is binding to any other site. The word allos or ALLOS is just Greek for other, okay? So it just means some other site on the protein that when you bind there, generally what will happen is a conformational change, making the active site less available, so that the regular ligand or molecule can't get in. So then you want to, I read this as lead every time. It's so hard for me to read this as lead compound for some reason. And I'm like, none of these drugs would have lead. But so if you're identifying a lead compound, usually what you'll do is you will run an assay, some sort of high throughput screening where you have this huge library of a bunch of type of small molecules, okay? They're all similar. They might have a couple of different side groups on them. Maybe one has a fluorine, maybe one has a couple nitrogens available, whatever it might be. And what you're looking for is you're trying to match a binding pattern to your target site. And sort of once you identify a lead compound, kind of getting a hit through this high throughput screening, then you'll go through a series of iterative steps to improve the affinity of your compound until you have sort of a drug you want to move forward with. Okay. So anti-cancer drugs are often targeting kinases. Protein kinases are enzymes that transfer a terminal phosphate of ATP to a hydroxyl group. Yes?
On the last slide, I was wondering what kind of factors do change the affinity in that sense?
Perfect. There's a slide 2 from this that will show literally you just kind of change some side groups on your lead compound and you check the affinity with binding assays again. And so the one that will come up for this Gleevec, this particular cancer drug, I'll populate it. It'll show like they tried some fluorine groups, then they tried some nitrogen groups. It's literally just kind of a...
So it depends on non-polar versus polar interaction?
It's going to be, there has to be a mix of both. You have to have both non-polar groups and polar groups. You want to try and match like a pattern of hydrogen bonds or positive and negative interactions. But in order to increase affinity, you want to put in more hydrophobic. So it's kind of a mix. Hopefully when it pops up, you'll see some, literally it's just some things they tried. Okay, so protein kinase, that's a great question though. How do you raise affinity, put on hydrophobic groups?